Hi, my name is Livy Norman, and today I'm going to be discussing my poster, which is titled Investigating the Role of CHRFAM7A in the Immune Response of Human Macrophages. I have no relationships with for-profit or not-for-profit organizations, and this program has not received any financial or in-kind support. My research within the context of the immune system focuses on the inflammatory response mediated by macrophages. Macrophages are a specific type of white blood cell that modulates the inflammatory response through the release of small signaling proteins known as cytokines. Cytokines allow macrophages to communicate with other cells to either increase the inflammatory response and recruit more white blood cells to the site of infection, or to decrease the inflammatory response through immunosuppressive action. There are numerous cytokines that promote inflammation, but for this project, we focused on tumor necrosis factor alpha, abbreviated as TNF-alpha, and interleukin-6, abbreviated as IL-6. The cytokine interleukin-10 was chosen for this project for the characterization of anti-inflammatory cytokine secretion by macrophages. Macrophages are known to express nicotinic acetylcholine receptors, abbreviated as NHRCs, which have been found to play a role in the inflammatory response. The gene we have focused on in this project encodes a nicotinic acetylcholine receptor that is found only in humans and has an unresolved function in the immune system. To elucidate the role of this gene, known as CHRFAM7A, in the immune response of human macrophages, we created multiple CRISPR-mediated CHRFAM7A knockout and overexpression cell lines in THP1 macrophages. The immune response of these cell lines was characterized by stimulation with the immunogen lipopolysaccharide, both with and without the NACHR agonist nicotine. The resulting immune response was measured using ELISA assays for the cytokines TNF-alpha, interleukin-6, and interleukin-10. To create CRISPR-mediated CHRFAM7A knockout and overexpression cell lines, specific guide RNAs were designed and cloned into lentiviral vectors. These vectors were transfected into HEK293T cells, which produced lentiviral particles encoding the guide RNA sequences and released them into the cell culture media, which was collected. Cas9 and dCas9 expressing THP1 macrophages were infected with CRISPR guide RNA encoding lentiviral particles, resulting in CHRFAM7A knockout and overexpression THP1 macrophages. The results shown on this poster are limited to the CHRFAM7A overexpression cell lines, as the knockout cell lines are still in the process of verification. Figure 1 demonstrates the relative mRNA expression of each of the CHRFAM7A overexpression cell lines for the genes CHRFAM7A, dCas9, guide RNA, and CHRNA7. Compared to the THP1 macrophage cell line expressing dCas9 and an empty guide RNA plasmid, which was used as a control, all three overexpression cell lines demonstrate significantly increased expression of CHRFAM7A. Based on the mRNA expression of the genes dCas9, guide RNA, and CHRNA7, the first overexpression cell line was chosen as the best candidate for cytokine analysis. This is because the first cell line was seen to have the highest mRNA expression of CHRFAM7A with the fewest off-target effects. Figure 2 demonstrates the immune response of the first CHRFAM7A overexpression cell line after exposure to the immunogen lipopolysaccharide, both with and without the NACHR agonist nicotine. Graph A shows that CHRFAM7A overexpression cell line had an increased LPS-induced TNF-alpha secretion compared to control. Graph B shows that there was no significant difference in LPS-induced interleukin-6 secretion between the CHRFAM7A overexpression cell line and the control cell line. Graph C shows that the CHRFAM7A overexpression cell line had a decreased LPS-induced interleukin-10 secretion compared to control. In conclusion, we were able to successfully create 3 CHRFAM7A overexpression cell lines as evidenced by panel A of figure 1. In addition, we observed that within the CHRFAM7A overexpression THP1 macrophages, there was an increased secretion of the pro-inflammatory cytokine TNF-alpha, no effect on the secretion of interleukin-6, and a decreased secretion of the anti-inflammatory cytokine interleukin-10. Overall, based on these results, CHRFAM7A overexpression has a pro-inflammatory effect on the immune response of human macrophages.